Welcome to Bioinformatics. I am Dr. Y.V. Rajesh, Department of Biotechnology, KL University. So in previous session we learned about a dynamic programming approach of pairwise sequence alignment. In today's session we will learn about heuristic approaches called as Fasta and BLAST. So you may ask me one question: why we are using dynamic programming approach? Because dynamic programming approach is called as the question is something like this: why we are learning heuristic approach or these algorithms when dynamic programming is the best algorithm? So there is a simple reason is there. The dynamic programming may be exact approach, but the real issue is time consuming. So actually basically we don't have much time and we want to get the results immediately, interpret the results immediately and get insights as soon as possible here. So time consuming approaches, the dynamic programming approaches, and they are not feasible for longer sequences. You have very long sequence. Suppose if you want to compare genomes like human genomes of 3 billions of bases with mouse genome another 3 billions of bases, the dynamic programming approach is not at all possible here. And dynamic programming basically computationally intensive. That means you need a lot of computational memory. Sometimes you require supercomputers, and we don't want to hang our laptops while doing some sequence alignment work. That's why we need another kind of approach, another kind of alternative approach, which should not compromise with the scoring methods, scoring of the alignments. At the same time, it should give the results very quickly. So those approaches are called as heuristic. So the meaning of heuristic is give solution in quick time. Answer may not be optimal, that means quick time, but the answer may be suboptimal. So it depends upon the user to choose that solution or not. So but most of the cases, so it is choosable only because it's a suboptimal approach. So heuristic approach, two algorithms are there, Fasta and BLAST. So the name then is it's a short form of fast adaptive shrinkage threshold algorithm. Fast adaptive shrinkage and threshold algorithm. And BLAST is basic local alignment search tool here. The idea is very simple. So instead of taking all the characters at a time, you take some specific characters called as k-mers or k-mer or word search approaches, then try to resolve the solution to the problem here. So first thing is find the runs of identical words. Three steps of the fast algorithm is there. First you find runs of identical words. So try to pick up the matching simple words in two sequences, and the identical runs of words you call them as tuples. Tuple n equal to, it can be 1, 2, 3 like this here. Basically we start from 2 here. So k tuple equal to 2. That means in DNA you have AA, AC, AT, AG, CA, CG will become tuple equal to 2. And when tuple is 3 here, you have AAA, AAC, AAG and AAT. So user will decide what tuple you want to take when you want to design his own concept here. But here the Fasta algorithm uses tuple of 3 for protein sequences here. So then first you take tuples, then after words are matching, if it is a protein sequence, use something called as scoring matrix. Scoring matrix we'll discuss later sessions, and scoring matrix, a method called PAM, percent accepted mutations matrix or point accepted mutation matrix. It's a lookup table. From that you try to see the characters and you replace the score with the score which is present in the matrix here. So rescore using PAM matrix. So that is the second step here. So assume that here my sequence A and B, which forms the two axes. For visualization only we are showing this one, which forms the axis here. So first pick up identical words here. Then after getting the identical words, then you rescore them using PAM scoring matrix. Then third step is you pick up the longer matching segments and you match them here. So third step is pick up longer matching things, okay, matching things, and you join longer matching segments and you join them using gaps here, and eliminate other things, short other things here. So during the process what we do is you pick up top 10 or 10 best words, matching words, 10 best longer matching words in two sequences here. So once this is done, then finally you join, we said about joining them, but after joining you need to provide some kind of score here. The score can be created using the dynamic programming approach, using Smith-Waterman algorithm. So using that approach, simply you provide the score for alignment here. So you pick up the 10 best matching words and you join them and score them using Smith-Waterman algorithm. So this is a Fasta approach. This Fasta actually has many applications, and you can perform Fasta for doing so many things here. A Fasta algorithm can compare a protein sequence with a protein sequence, so that is called as a simple Fasta, or a DNA sequence with a DNA sequence, both are called as a simple Fasta here. So DNA to DNA, protein to protein comparisons. And T Fasta, T indicates translated. T Fasta compares your protein sequence to a DNA sequence library. So basically, although it is a pairwise sequence alignment, the pairs of sequences, so what we are going to do is we have a library or data bank where millions of sequences are there. So you have one single sequence, our single sequence is compared with millions of sequences in the database one by one, so as a pairs here. So a query protein sequence to a DNA sequence library, if you are doing that, that is called as a T Fasta here. So in this case you translate your DNA sequence library into all 6 open reading frames. And another flavor of or version of Fasta is there, FastX and FastY. So when your sequence is a DNA sequence, you convert that one into all 3 different reading frames, forward strands, and another 3 frames on reverse strand, and compare with a protein sequence database, that is FastX and FastY. And TFastX and TFastY here. So what we do is you will compare a protein sequence to DNA sequence database by translating each DNA sequence into all 6 possible reading frames. So 4 different flavors of Fasta algorithm is there. So we'll learn about the Fasta algorithm in detail while doing Fasta lab experiment. So we'll take a sequence, a novel sequence, and we'll try to compare whether how much percent of sequences matching with real the database sequence here. So thank you very much for listening to this session. So in next session we'll learn about BLAST algorithm. Welcome to another class.